Hey guys, my name's Ashley, and today I'm kind of here to re-summarize Dr. Volley's BMB 401 Lecture 35 on replication in both bacterial and eukaryotic systems. So obviously Lecture 35 had a lot going on, and while I felt all the content was there, I felt it was a bit disorganized at times. So this is kind of my attempt to reorganize things and then re-explain them. A little bit about me. I am a junior right now. I'm majoring in genomics and molecular genetics as well as nutritional sciences. I'm also currently an undergraduate learning assistant for the Lamb-Briggs chemistry team. Go chemistry, but most importantly, assuming you are my target audience, I am your classmate, a fellow student in BMB 401. That being said, I'm certainly not an expert, and I also didn't really do a practice run of this before hitting record. So it's kind of just be pretty informal, me walking through my notes with some figures I have pulled up. And yeah, so we're gonna go ahead and get started. So the beginning of the lecture kind of sets us up for what we need to know about everything we're gonna be told. We wanna know what's happening, when, where, how does this process begin, what's required, and what's the directionality of these enzymes, which we'll all know in due time, but kind of just to set us up for what we're looking for. For our first set of figures, I just kind of have E. coli versus human cells. Obviously, they're very different, so it makes sense that they'd have different things going on. Okay, so we know that replication is DNA synthesis and preparation for cell division, which makes sense because if you're going to make two cells out of one, you probably need more DNA than what you started with. We also know that replication is semi-conservative, so I have this figure here to help us out. We don't really need to pay attention to B and C, but more focus on A here. B and C were other models of replication that were theorized but turned out to be wrong. So semi-conservative replication kind of works as where you have your parent strands, and then after that first replication cycle, each cell, the parent cell and the daughter cell, are going to have one of that original strand and one new strand. If you take it farther and go to the next generation, you'll see that one of those daughter cells has two completely new strands and one has a new strand and original strand, kind of proving that this is semi-conservative replication. You don't really need to understand why it's semi-conservative as much as you just need to understand that it is, and we know that we are keeping an original strand and synthesizing a new one. If you're looking for more information about how we discovered that DNA replication was semi-conservative, you can check out this experiment by Meselson and Stahl. Very cool, covered in MMG 431. Shout out to Dr. Waters. Moving on, so we talked about how parent DNA synthesizes a daughter strand and a daughter strand from the parental template so that the parental cell keeps a parental strand and has a daughter strand so that both the parent and the daughter cell end up with one of each. Okay, so obviously replication in bacteria, or I guess replication in prokaryotes, is going to be different than replication in eukaryotes. And for our two model organisms, we're going to be using E. coli for prokaryotes and humans for eukaryotes. So even though they're pretty different, they do require similar components. For one, you need a DNA template strand, which makes sense because if you're going to make DNA, you probably need to know what that DNA has to say. We also need ribonucleotides to form RNA primers, which we'll get to in a bit, so don't worry if you don't really know what that means. But what exactly is a ribonucleotide? And then looking down here, what is a deoxyribonucleotide? Okay, so we know that ribonucleotides and deoxyribonucleotides work together or work with other components to make nitrogenous bases, which you can see it down here. So the difference comes here on this 2' position. On a ribonucleotide, we have this hydroxyl group hanging out here, whereas on the deoxyribonucleotide, we don't have this here, which is why it's called deoxy. It's without the oxygen. Generally, RNA is a little bit more reactive than DNA, which is probably why we see the gradual evolution over time of an RNA world into a DNA world. But we don't really need to worry about that, but that is why those two exist. One probably started with RNA and then eventually found its way to DNA. Okay. So we need ribonucleotides. You must make RNA before you make DNA. We'll get to that in a little bit, but basically you need to lay down RNA in order to allow DNA polymerase to attach. But don't worry about that for right now. You also are going to need polymerase enzymes and accessory enzymes and proteins. Well, yeah. Okay, but obviously there's differences as well. As far as a bacterial genome goes, E. coli has one circular chromosome, so I kind of have that right here, and there's around 4.6 million base pairs. Each circular chromosome has one replication start site. And we know that cell division is based off of cell size, which I'm sure you've heard before. We want a high surface area to volume ratio, which is typically why we see cells being spherical and very small, because we want that higher ratio. However, each different bacterium has its own optimum size, so they're going to be a little bit different across. When the size is passed, it's time to divide. As far as the eukaryote genome goes, humans have 46 linear chromosomes, so a lot more than that one circular chromosome, and we also have 6 billion base pairs. So obviously that's a lot more DNA to copy, so there are multiple replication start sites. And we can kind of see this in this figure here. We have a start site, one here in the circle here, and then multiple on the linear chromosomes. Okay, and also in eukaryotes, we see cell division is highly regulated, including cell type and developmental state and intracellular signals and extracellular signals. So all these things can influence what cell division is doing. Okay, so moving on to replication in E. coli. So this is going to be replication in prokaryotes. We are going to start with initiation. Just kind of a key point here, DNA synthesis cannot initiate without a primer. We mentioned a little bit ago that we need that primer to attach. Okay, so initiation is beginning in the origin of replication, which is commonly abbreviated as OREC. And we can kind of see this in this figure here. This is where replication is going to start. I hope you can see my mouse. I didn't really check that before I started. Anyways, the OREC locus is a specialized region, and it's around 245 base pairs. OREC itself is a DNA sequence that consists of 2 regions. We have a tandem array of 3 AT-rich consensus sequences. So I got this figure from the internet, and well, I don't know exactly where this came from. We're certainly seeing a lot of T's that would bond to a lot of A's. So why is this DNA easy to melt here? Well, we know that when an A and a T base pair, there are 2 H bonds being formed, and which is weaker than a G and a C pairing, which has 3 H bonds. It's really important that you understand that these are not covalent bonds. I felt that in this lecture and in previous lectures, we've kind of skipped over intermolecular forces, but when we are talking about H bonding here, we are referring to intermolecular forces as opposed to intramolecular forces, which would be like covalent and ionic bonds. So I have a little picture here. We can see A and T, H bonding between that hydrogen and that nitrogen, right? That makes sense because this nitrogen is electronegative. And over here, we can see this hydrogen is partially positive because it's attached to an electronegative nitrogen. So this is partially negative, this is partially positive. That's why we're seeing intermolecular forces. Over here, we can see there are 3 hydrogen bonds. So it makes sense if there's more intermolecular forces, we know it's going to take more energy to disrupt. So that's why we're seeing that this tandem array is a little bit easier to melt just because it has a lot of those AT base pairing as opposed to GC. Okay, what's also happening in this OREC is a DNA A protein binding site. So eventually DNA A is going to be able to come in here. Okay, so, oh, I forgot an A there, I'll add that. DNA A is an enzyme and it uses ATP. It opens DNA strands at the origin of replication. So the first thing that's going to happen is DNA A binds to OREC. It uses ATP hydrolysis to power the separation of DNA and separation of DNA strands at the ORE locus. Okay, so then that DNA A is going to unwind the DNA beginning at the AT-rich region. So it's going to start unwinding. I forgot where I was reading. Okay. Anyways, a single-stranded binding protein is going to keep these strands separated. And we need these here to prevent higher order structures such as stem loops or, as we'll see later, supercoiling. And we want to make sure that this DNA doesn't re-anneal. So to help us out with that, we're going to form this prinosome. Okay, so in order to do this, first we need to make a short stretch of RNA. So DNA A is going to unwind the DNA, just like we talked about, at the origin of replication, and it's going to recruit 2 proteins called DNA B and DNA C. And I kind of like this picture. It's kind of simple. We don't really talk about DNA E, so don't worry about that. But you can see DNA B coming in here. Okay, so DNA A is a multimer that contorts the origin of replication, right? We talked about that because it makes sense. It's going to contort that origin so that it can open up. DNA B is a DNA helicase that further unwinds the DNA. So the DNA A kind of got it started, and now the DNA B is going to take that and run with it. It consists of 6 individual subunits that comprise a ring-like structure. Cool. It binds to the DUE, which is a common abbreviation for DNA unwinding element, and unwinds DNA to increase that single-stranded region. So we want more single-stranded DNA so that we can have access to it and start replicating on it. Okay, and it's also associated with the primase, and this primase is called DNA-G. All right, so DNA-C, it got called over by DNA-A earlier. It's going to help DNA-B get onto that DNA at the origin of replication. And DNA-C is also going to help open up that ring-like structure we saw under DNA-B. Okay, DNA-G is an RNA primase that is transiently bound to DNA-B and synthesizes that RNA primer that we need to attach DNA polymerase. We're almost there. We keep mentioning DNA polymerase, and we're going to eventually define it in a hot second. Okay, so only after that RNA primer is down can that DNA synthesis go. We've already mentioned that a couple times. You need RNA first. Okay, so DNA is going to be synthesized in 2 directions, and directionality matters for sure. However, this is kind of a point in the lecture where she uses the words, Dr. Fowler uses the words top and bottom. Definitely don't do that. You never want to be using the terminology top and bottom, because really there's nothing saying we can't take whatever picture you were looking at and rotate it upside down. It would still work perfectly fine. So instead of using the terms top and bottom or left and right, we really want to be using the terms 3' to 5' or 5' to 3'. That's going to tell us how you're reading this DNA and which way it's facing. Okay, so 2 chromosomes made up of DNA-B and DNA-G found on the chromosome. Okay. DNA is read 3' to 5', and DNA is synthesized or built 5' to 3'. You absolutely have to know that difference, and honestly, it's going to be a struggle if you don't. Okay, so the place where the DNA is separating is called the replication fork. There's going to be 1 to the left of this ORAC and 1 to the right. I thought I had a figure here I wanted to show. I don't know where it went. Oh, okay. Oh, and here I have don't say, yep. Do not say top or bottom or left or right. Okay, fine. Anyway, so there's going to be 1 to the right of ORAC and 1 to the left. So we can kind of see, I like this one here because it shows that middle line of where it started and that the fork movement is going to the left and the right, as seen by these arrows right here. Okay, so 1 to the left, ORAC, 1 to the right. This helps to recruit the main replicative enzyme, which is going to be DNA polymerase 3. Now, Dr. Fowley uses a couple different terms for this enzyme. I will be calling it DNA polymerase 3 for the entirety of this lecture. I really recommend you do so too, because I don't know, there's like polymerase 3, there's DNA poly 3. I just like to call it DNA polymerase 3, or if I'm typing it, DNA P3. Okay, DNA P3 is part of what we call a replisome, and there's going to be 2 of these replisomes happening here. And this replisome is going to consist of 3 main parts. So we're going to have a beta-2 clamp, which you can kind of see here. I have a bacterial fork and a eukaryotic fork, but we're looking at this bacterial fork. And okay, beta-2 clamp holds DNA polymerase 3 to the DNA strand. Okay, so they're taking DNA polymerase 3, and we're going to make sure it stays there. DNA polymerase 3 itself has 3 parts. The alpha, and I have a picture here, so I guess if you want to look at just DNA polymerase 3, you have an alpha, which is going to be the catalytic portion, the part that's reacting. Theta, we don't really know. And epsilon has exonuclease capabilities. It can take off nucleotides when DNA polymerase 3 makes it a mistake to give it another chance. So yes, DNA polymerase 3 is capable of making mistakes. It can bond the wrong base pairs, it can set down the wrong base pairs. So we want to have repair mechanisms in case something goes wrong. I think we're definitely getting to more DNA repair mechanisms later, but for right now, DNA polymerase 3 has an epsilon part that gives it the ability to fix itself. And then the last part of our replisome is going to be the clamp loader, which opens and closes the beta clamp to get it on. Here it is, it's a complex here. And then up here, you can see the clamp loaders right there. Kind of abstract, kind of abstract in these images. I would say it's probably more important that you know what they do as opposed to how it looks in a 3D diagram. Ideally, you want to know both, but I'm having a little bit of difficulty myself. Okay, so we know that one of these ribosomes is going to go to the left and one's going to go to the right. Each synthesizes 2 strands of DNA at the same time. Again, I have another reminder here, do not use top or bottom or left or right. You really want to use 5' and 3' ends of nucleic acids. And that's kind of why we use that, is because this 3' site right here, that's what we can build off because it has that oxygen, which is capable of reacting with other things. We can't build off the 5' end because we have this phosphate backbone, so it's hard to get something to react with that. Okay, finally getting to replication. So there are 2 replication forks, 2 strands of DNA in each. Okay, lots going on. One of these strands is going to be facing the right direction. And what does that really mean, right? What is the right direction here? It's going to be laying anti-parallel to its template strand, right? Because DNA lies anti-parallel to itself. One strand goes 5' to 3', the other is going to go 3' to 5'. Okay, so this strand that's going to go the right direction is going to lay down correctly on its template strand. And it's going to be able to synthesize because it's facing in a direction where the right way it has to go is just 3' end. So since we've synthesized 5' to 3', it's going to be able to build off of that 3' site. And we talked about how that was reactive. Of course, I don't know where the picture went. Whatever, we talked about how you were able to build off that 3, it's literally right here. We talked about how you're able to build off this a lot more easier than this 5' site right here. Okay. So then the other strand is going to be facing the wrong direction. And we call the one that's facing the right direction the leading strand, and the one that's facing the wrong direction the lagging strand. So we can see right here in this strand that's facing the right way, look at the template strand here. It's going to go 5', and so we go up over here. We know this must be 3'. So then this leading strand is going to attach 5'. It can synthesize 5' to 3'. Okay, but now there is a strand that is facing the wrong way. So it's laying anti-parallel to its template strand, but it needs to build DNA where its 5' site is. Okay, so it would need to build off that 5' site to go the right way, to be anti-parallel to its template strand. So it's going to build these baby fragments in order to override this, because it can't go 3' to 5', so it's just going to do a bunch of baby 5' to 3' to make up for that. So I kind of have a picture here to help us out with that. You can see our parental DNA is 5' to 3'. Our leading strand can go the other way, no problem. But since this parental strand right here is 3' to 5', it would have to, our DNA fragments would have to, or I guess our template strand right here is going to make the strand we are trying to encode it would have to go towards the center of this fork, which is obviously not going to work because that would be going 3' to 5', and we can't build that way. Whew, just a little bit tricky. Okay, so we want to make these little baby fragments so that we can synthesize DNA on this other side, because we need to do DNA on both sides. Okay, so these are called Okazaki fragments, and they are baby chunks placed one after another so progress can be made in the opposite direction. For each Okazaki fragment, we have to synthesize an RNA primer so we can place them down. We talked about how we have to put RNA down first to get any DNA. So just like before, DNA B, which is DNA helicase, is going to open up the replication fork, and DNA G is attached. And I kind of have a summary image down here. We see eventually an Okazaki fragment here. Of course, I don't have the right pictures to show everything. Whatever, we'll just keep going. Okay, so again, DNA B helicase is going to open up that replication fork, and DNA G is attached. And we saw before that DNA G starts to synthesize that short RNA primer. And then after that RNA primer exists, DNA polymerase 3 can attach, and it synthesizes that short stretch of DNA. Okay, so once this DNA is synthesized, the beta clamp releases, and the DNA polymerase 3 can float away and go do something else. The cycle is repeated until the lagging strand is synthesized. Okay, cool. So then DNA polymerase 1, not 3, can take nucleotides out going 5' to 3'. So it kind of acts like a snowplow to remove all these leftover RNA primers from the lagging strand and replace it with DNA. Cool. Okay, and then after that, DNA ligase is able to seal the breaks in the DNA. Awesome. So here is kind of a summary image here. We can see the Okazaki fragments hanging out here. We've got RNA primers. We've got DNA ligase to seal the gaps. We've got DNA polymerase to help synthesize that DNA. Lots of good stuff going on, and there's lots of good figures on Google as well. So after that, we kind of do a little bit of summary. You need the following for DNA replication. DNA A uses ATP to separate DNA at the origin of replication. DNA C to recruit DNA B. DNA G is that primase, so lay down the RNA primers. Our main synthesizing polymerase is going to be DNA P3, DNA polymerase 3. It synthesizes both the leading and the lagging. DNA polymerase 1 comes and removes those RNA primers from the Okazaki fragments and fills with DNA. Cool. Okay, so DNA ligase seals the breaks on the lagging strand, and the parent and the daughter cell each get one parent strand and one daughter strand. These two strands become unlinked through an enzyme called topoisomerase 2. Okay, I guess that's the next thing we're diving into. Just make sure got everything up here. Cool. So DNA polymerase 1 is removing the RNA primers. Cool. And replacing it with DNA. Okay. All right. So we know that in eukaryotes, the replication of the genome is a tightly controlled part of the cell cycle. And we know the cell cycle is, we have stages G1 for growth, S for DNA replication, G2 for air repair, M for cell division, and G0 is exit from the cell cycle to go do whatever it wants. Cool. So we know this is complex and highly regulated, this replication. There are many origins of replication per chromosome, right? We talked about needing a lot more origins of replication to synthesize that 6 billion base pairs. And each used once per cell cycle. Awesome. So the origin of replication complex. Okay, and we see this during the G1 phase. So this origin of replication complex is, and we can see, can we see it on here? Well, maybe the subcomponents. Okay. Unwinds DNA a little bit and recruits these licensing factors. All right. So it's going to be using ATP hydrolysis, and it's similar in function to DNA, right? Because we saw DNA going in and unwinding that DNA a little bit. And then this origin of replication complex is going to recruit CDC6 and CTD1. I have never heard of these before, so I don't know a whole lot about them. Probably just need to know their players and they're happening. And we know that these licensing factors, which are similar to DNA B and C, these players right here, the important part is that they're loading MCM27 helicase. And we don't really need to know what MCM27 stands for, mini chromosome maintenance protein helicase. We just need to know that it's similar in function to DNA B. It's unwinding that DNA at the replication fork. So 2 plays for each origin of replication, one to the left and one to the right. Many are loaded on the chromosome prior to the S phase. Coolio. Okay, so just kind of drawing comparisons between these players for eukaryotic replication and players for prokaryotic replication. Awesome. Okay, so we got this DNA polymerase A primase complex, alpha primase complex. We can kind of see on this eukaryotic picture here. And it synthesizes RNA primers and replaces this with DNA. Okay, so that is similar to what we saw in DNA G and DNA P1. And again, like we saw in eukaryotic replication, DNA P1 is the one going in and synthesizing off these RNA primers for these short strands. Yeah, and there's still small pieces for those lagging strands because even in eukaryotic replication, we're still encountering that same lagging strand difficulty. Awesome. So all of our polymerases, cool. So we have DNA polymerase delta, which synthesizes our lagging strand. We have DNA polymerase epsilon, which synthesizes the leading strand. And both are similar in function to DNA polymerase 3. So both of these together can kind of do the same thing that DNA polymerase 3 does. There are two main polymerases as opposed to one in E. coli. And these are held in place by a sliding clamp, proliferating cell, nuclear antigen, just knowing it's pCNA. We can see on this picture here, I don't understand these figures super well because I just pulled them from the internet, but doing the best we can here. Okay, and this has a similar function to the beta 2 sliding clamp, right? We saw was able to help out in prokaryotic replication. Okay, so human chromosomes are linear, and that means they have these things called telomeres at the end of them. And this telomere is a portion of the DNA that consists of repeats that are folded on each other to protect the DNA. So we can kind of see in the figure here, they're folding on each other and they're wrapping around. They're protecting the end of that DNA. Telomeres contain tandem repeats of a 6 nucleotide sequence, GGG-TTC. Okay, we can kind of see lots of repeat sequences here. Cool. On one strand, this region is longer than on the other, and the looping structure it forms protects the telomeres against exonucleases. Cool, so they can't be cut away, they're here to stay, and they're going to protect that DNA. These telomeres must also be replicated so that DNA can continue to be protected. So telomerase is a specialized reverse transcriptase that carries its own RNA template. Okay, so reverse transcriptase, we're going from RNA to DNA. So it's finding and it's binding to that telomere. It's extending and synthesizing DNA to make a telomere, and then it folds onto itself to make the ends of the chromosome. Cool. We can see here this one strand is longer than the other, so it makes a loop. And here we're seeing telomerase come on, it's elongating, and then it's folding onto itself. Cool. Again, not a super expert at this, but we're doing the best we can. Okay, so DNA can become overly coiled because lots of unwinding is happening during all this replication, so there's a lot of things happening. We need to be able to take care of this. So topoisomerases break either one strand or two strands of DNA to release this tension. Some put supercoils in, which is a higher order of folding that we talked about earlier, and some take these supercoils out. Generally, there is a turn in the DNA helix. I think in lecture 34, we talked about major and minor grooves. There's a turn every 10.4 base pairs, sorry. So for example, if something was 260 base pair long, there'll be around 25 turns happening for that entire sequence. So the number of turns you have for a sequence are called your linking number. So if you had 104 base pairs, you'd have a linking number of 10. If you change the linking number, you introduce supercoiling. Okay, so the first type of supercoiling we're introduced to is a RIF, or I think it's called. And it's the number of times a double-stranded DNA helix crosses over itself, like the infinity signs. I kind of have this here. The RIFs are like here and here. Okay, and then we also have a twist, which is the number of times the DNA twists around itself, which is different from the linking number because the linking number is one strand going around another. Okay, so the linking number is the twisting of two single-stranded DNAs to make that DNA helix. And we know that the twist is different from that because that's two double-stranded DNAs are linking around each other. So a little bit different. And we know that twists plus these RIFs equal a linking number. So this is your equation right here. As far as I can gather from the lecture, she's just going to give you the numbers, and she'll also tell you whether it's going to be negative or positive, and then you just have to solve for x. So she gives an example of there are two negatives, so we have a -2 for here, and a linking number of 10, and we can just solve that for 12. Cool. Okay, I think we're getting there. There are two major classes of topoisomerases. So topoisomerases change the linking number by making breaks in the DNA backbone, adding or subtracting turns. Okay, cool. So we've already learned that's a thing, and now we know what's going to do that thing. So topoisomerase 1 makes a break in one strand of DNA and changes the linking number by 1. Very convenient they got it right and named topoisomerase 1 for when it changes the linking number by 1. So I took the figure from the slide here just because I'm not super familiar with topoisomerase, and also that's kind of a cool looking structure, so why not? For this, you do not need ATP because it's using the existing tension in that DNA sequence to add or subtract linking number. Okay, it's going to use the catalytic tyrosine to break the phosphodiester backbone, and that's a transesterification reaction. So just know that tyrosine's holding that one strand while topoisomerase 1 can unwind the other strand, so it's kind of just holding it out of the way. And then once topoisomerase 1 has unwound that DNA, the tyrosine releases the strand and the bond is allowed to form again, and that bond being the phosphate backbone. Topoisomerase 2 makes a break in conveniently 2 strands of DNA and changes the linking number by 2. So this cuts double-stranded DNA as opposed to single-stranded DNA. It also requires 2 molecules of ATP, so it's not using existing tension. It's going to bring in its own energy. The DNA to be broken is called this G segment, so it enters into this gate here. It binds, the DNA is broken. The T segment, I guess, is allowed to go through this break. The DNA breaks are repaired. ATP is hydrolyzed to release the DNA, and the process can start again. Cool. So this has gone through, and they end up like that. Looks a little different than before. Over here. Cool. That's the end of lecture 35. I think that was an okay explanation. Please let me know if you have any questions. And also let me know if you want to see another one. I haven't watched lecture 36 and beyond yet, but I am assuming they're also about DNA. So yay. Yeah, thanks for watching. I guess I'll end the recording now. Bye-bye.